Good morning, everyone. Let us introduce ourselves.
My name is Nathaniel Chochai. My student number is 661055.
Hello, my name is Prashapon Khemdi, and my student ID is 661090.
My name is Pimalan Jirakom, student number 661404.
My name is ธนิตประกอบแก้ว Student number 661419.
My name is Shishanok Lohita, student number 661421.
My name is รัตนันท์ ศิวรสาร Student ID 661422.
Let's move on to topic 1, the importance of working in a microbiology laboratory. Nowadays, microbiology plays an essential role in human life. Microorganisms such as bacteria, viruses, fungi, yeast, and protozoa have a significant impact on ecosystem, human health, industries, and the environment. Although they are invisible to the naked eyes, their presence influences the world in both positive and negative ways. Some cause diseases, while others are highly beneficial used in medicine production, food processing, and wastewater treatment. The microbiology laboratory is the core facility where scientists examine and understand these microscopic organisms. Working in this lab is not just about performing basic experiments. It is the foundation of scientific discovery that can directly influence lives. For example, in the medical field, microbiology labs are essential for diagnosing infectious diseases, testing bacterial resistance to antibiotics, and identifying new pathogens. This discovery leads to the development of effective medication and vaccines. In the food industry, microbiology labs ensure food safety by testing for contamination by harmful pathogens, such as Salmonella or Listeria, which are common causes of food poisoning. These labs also play a key role in cultivating beneficial microbes like lactobacillus used in yogurt and probiotic production. In environmental science, microbiology labs assess water quality, monitor pollution, and analyze soil microorganisms to support sustainable agriculture. Moreover, the microbiology labs is a critical training ground for scientists, students in health science, and biotechnology researchers. It requires a deep understanding of biology, chemistry, laboratory techniques, and data interpretation to perform experiments and evaluate outcomes accurately. Understanding microbes is a vital tool for public health preparedness, climate change mitigation, and sustainable resource management. In this sense, the microbiology labs is not just research space. It's a front line for protecting human life and the planet. It is the origin of countless innovations that continue to improve our world.
Let's move to the topic 2, essential equipment and tools in a microbiology laboratory. Working in a microbiology laboratory requires specialized equipment due to the nature of work, which involves handling microorganisms that are invisible to the naked eye. Proper tools ensure accurate experimentations, minimize contaminations, and protect both the researcher and the environment. The first essential tool is the microscope. There are several types, include the light microscope, commonly used for observing stain or live microbial sample, and the electron microscope, which offer much higher magnification and resolution. The latter is used in advanced research for observing fine structural detail of microbe at the nanometer level. Another crucial piece of equipment is the biosafety cabinet. This is a workspace with control airflow designed to protect users from exposure to harmful microorganisms and prevent contamination of sample. This is especially vital when working with pathogenic organisms such as virus, TB-causing bacteria, or genetically modified microbes. The autoclave is a key tool for sterilizing reusable laboratory equipment. It uses pressurized steam at high temperature, usually around 121 Celsius, to effectively kill all forms of microbial life, including spores. Autoclaves are also used to safely decontaminate biological waste before disposal. Let's move to the next page. Next is the incubator, a device used to grow microorganisms under controlled environmental conditions. It maintains an optimal temperature, often between 35 to 37 Celsius, for the cultivation of bacteria or fungi. Some advanced incubators can also control humidity and oxygen level. Additional tools used daily in microbiology lab include pipettes for precise measurement and transfer of liquid, test tube and beaker for mixing and storing samples, petri dish used to culture microbial colony and agar media, inoculating loop for transferring microbe and safely accurately, centrifuge used to separate components of a liquid such as cell, DNA, or protein. All equipment must be properly sterilized before and after use. Instruments like pipette and incubator must be regularly calibrated to ensure reliable data. Safe operation require proper training, meticulous technique, and careful handling of material. Finally, laboratory safety infrastructure is critical. This includes chemical storage cabinet, hand washing station with antimicrobial soap, personal protective equipment such as gloves, lab coat and safety goggles, and biohazard waste container. These elements ensure the microbiology lab remain a safe and efficient environment for scientific investigation.
Move on now to topic 3, procedure and method for cultivating microorganisms. Microbial cultivation is one of the most fundamental procedures in microbiology. It enables researchers to study microbial behavior, growth patterns, and biochemical characteristics. Proper cultivation techniques are essential for obtaining accurate data for future analysis, such as species identification, antibiotic resistance testing, or the production of bioactive compounds. Before beginning the cultivation process, scientists must first prepare culture media, which serve as the nutritional source for microbial growth. There are many types of media issued to different microorganisms. For example, nutrient agar used for general bacterial growth, support agar designed for culturing fungi and yeast, McConkie agar are selective and differential media used for gram-negative enteric bacteria. All media must be sterilized using an autoclave to prevent contamination. The sterile media is then poured into petri dishes under sterile conditions, usually inside a biosafety cabinet, to maintain a clean and controlled environment. Next, researchers must collect microbial samples, which may come from water, soil, food, or clinical specimens. such as blood, urine, or sputum. The collection process requires aseptic technique to prevent external contamination. Once samples are obtained, microbiologists use inoculating loops or needles to transfer microbes onto the culture media using variant methods. 1. The stick pen method is used to isolate individual colonies. 2. The propane method is for quantifying viable microbes. 3. The spread paint method evenly distributes the sample across the plate surface. After inoculation, the petri dishes are placed in an incubator which is set to the temperature suitable for the target organism, typically 37 degrees Celsius for human-associated bacteria, or 25 to 30 degrees Celsius for fungi and environmental microbes. After the incubation period, researchers observe the colony that have formed. Each colony may differ in shape, color, size, textures, or even smell very cool about the organism identity. The colony are then analyzed further using techniques like gam staining, biochemical tests, or antibiotic susceptibility testing. Every step must be carried out with attention to sterility and safety to avoid contamination and ensure the integrity of the result. After experiments are complete, all used material must be properly sterilized, usually by autoclaving before disposal. Therefore, cultivating microorganisms is not just about growing tiny organisms. It's a process that requires scientific knowledge, skill, and precision. Done correctly, it becomes a power tool for discovery and understanding in the world of microbiology.
Now let's move on to topic 4, identification of microorganisms. Once microorganisms have been cultured, the next crucial step in a microbiology lab is microbial identification. This involves a series of physical, biochemical, and genetic techniques to determine the exact species or strain of the microorganism. The purpose of identification may vary from diagnosing infection to ensuring food safety, monitoring industrial production, or studying ecology system. The identification process often begins with examining colony morphology on an agar-plate. Scientists look at things such as color, shape, age, elevation, texture, and odor. These characteristics provide early clues. For example, Staphylococcus aureus typically form golden yellow colony, while Pseudomonas aeruginosa may produce a grape-like smell. Next is microscopic observation, particularly using gam staining, which divides bacteria into two main categories. Gram-positive bacteria have thin cell wall and appear purple under the microscope. Gram-negative bacteria have thinner wall and appear pink or red. Gam staining also reveals the shape of the bacteria, such as cocci, bacilli, and sphaerella, which help narrow down the identification. After morphology and staining, scientists perform biochemical tests to analyze the metabolic properties of the organism. This includes catalase test to detect catalase enzyme activity, oxidase test for detecting cytosol C oxidase, sugar performation test to check whether the microbe can break down certain carbohydrates. Additional tests like urease, indole, citrate, and triple sugar ion. These results are interpreted collectively to identify the microbial species such as E. coli, Salmonella, or Klebsiella. In recent years, modern molecular technology have revolutionized identification methods. DNA sequencing, especially 16S RNA gene analysis, allow for precise classification down to the species level. PCR polymerase chain reaction enable rapid and accurate detection even with small sample. Multi-top mass spectrometry analysis the protein fingerprint of an organism and identify it within minutes. In addition, many labs use commercial identification kits such as the API system or Vitech, which automate the testing and interpretation process, making identification faster and more reliable. Microbial identification play a vital role in clinical diagnosis, help doctors prescribe the right antibiotic and combat antimicrobial resistance. In the food industry, it ensures products are free from harmful pathogens, and in research, it supports discovery about microbial behavior, interaction, and potential benefit to health or the environment. In conclusion, identifying microorganisms is more than just assigning a name. It is a critical step toward understanding the invisible role of microbes and applying that knowledge safely and effectively in real-world context.
Move on now to topic 5 Laboratory Safety in Microbiology. Laboratory safety in microbiology is of most important because researchers work with microgenetics, some of which can be harmful to humans. Certain pathogens can be cause disease or spread easily if safety protocols are not strictly followed. Failure to do so many result in contamination, infection, or unintentional release of microbes into the environment or another people. Before work in microbiology lab, researchers must undergo safety training and understand laboratory regulation. One of the core concepts is biosafety level, classified with range from level 1 to level 4. Most endemic lab operate at BSL1 or BSL2, susceptible from large hazard microbes. However, for highly infection agents like Mycobacterium, tuberculosis, or Hypertherbi virus, BSLT certification are required. A fundamental rule is the use of personal protection equipment PPE including laboratory coat or gown, disposable gloves, surgeon mask or N95 respirator, safety glove or face shield. Their protection against during contact with infection, biological material, or work environment specimen or culture should be conducted inside biosafety cabin equipped with HEPA filter to provide the release of microbes into the air and ensure it's a still work zone. It is crucial to always practice aseptic techniques such as disinfection the workspace with 70% alcohol before and after experiment, framing including loop before and after use, avoid unnecessary talking during process to recruit information. Proper disposal of biological waste into designated biohazard bag followed by altering cape before disposal. If accident occurred, such as spill, splashes, or needle stick injured, they must report immediately. Emergency procedure include wound cleaning, disinfection, as modeling for protect contamination or exposure. Researchers should also understand how to safely handle chemical by reading MSDS material safety data sheet, storing chemical properly, separating flammable from oxidizer, and dispose of expiry agent safely. Safety also involves maintenance of lab equipment such as ensuring autoclave function correctly, regularly calibrating incubator and refrigerator, and installing sample in zero-calorie laboratory container. Lastly, personal discipline play a major role. Eating, drinking, or using mobile phone should never occur in the lab. Hand must be washed after every task, and contaminated material must not be taken outside the lab without proper sterilization. In conclusion, microbiology lab safety is not solely about equipment or rules. It relies heavily on responsibility, discipline, and awareness of individuals. With proper safety practice, research can be conducted effectively while protecting people and environment.
Let's move on to the last main point, which is disposal and decontamination of microorganisms after use. After completing any microbiological experiments, one of the most critical steps is the safe disposal and decontamination of microorganisms. This process helps prevent the spread of potentially harmful microbes and minimize the risk to lab personnel and the surrounding environment. Microorganism disposal must follow institutional biosafety standards and may include several methods. First, thermal decontamination. The most common and effective method is using an autoclave. It sterilizes materials using steam at 121 degrees Celsius under 15 psi of pressure for 15 to 20 minutes. Contaminated items such as petri dishes, test tubes, or biological waste are placed in biohazard bags before being autoclaved. After the process, items can be safely disposed of or sent for further waste treatment. Second, chemical disinfection. When heat sterilization is not feasible, such as for surface, bench tops, or sensitive equipment. Chemical disinfectants are used, including 20% alcohol, sodium hypochlorite, glutaraldehyde. These agents destroy microbial cell walls and metabolic functions. Proper PPE must be worn when handling chemicals to ensure safety. Third, biological waste disposal. Contaminated materials such as cotton swabs, gloves, used culture plates or tubes should be disposed of in clearly labeled biohazard bags. After autoclaving, some waste may be handled like regular trash, while others must be sent to a certified hazardous waste facility. Fourth, disposal of contaminated liquids. Liquids used during culturing or experiments that contain microorganisms must be chemically disinfected or autoclaved before being safely poured down the drain, only if permitted and after initial treatment. Fifth, record keeping and monitoring. In regulated labs, it is necessary to document the disposal process. This includes recording the person responsible, the autoclave cycle data, and the results of sterility indicators. 6. Decontaminating work areas. After each experiment, lab benches and nearby surfaces must be cleaned with appropriate disinfectants. Hands should always be washed thoroughly, and tools should be disinfected or autoclaved to prepare the space for future use. In summary, proper disposal and decontamination of microorganisms is not merely about throwing things away. It is a carefully regulated process that requires clear procedures, responsibility, and attention to biosafety. To ensure that our research activities do not pose risks to human health or the environment. I hope this presentation has provided some useful information. Thank you for your attention.
We'll start by the importance of working in a microbiology laboratory. Then we'll look at essential equipment and tools in a microbiology laboratory. Next, we explain about procedures and methods for cultivating microorganisms. Later, we showed you about how to identification of microorganisms. After that, we would like to deal with laboratory safety in microbiology. And finally, I will discuss about disposal and decontamination of microorganisms after use.